Excretion is the elimination of substances, usually wastes, from the body. Excretion usually starts at the level of the cell. For example, one of the waste products of aerobic respiration is carbon dioxide. So the cell excretes carbon dioxide. It's picked up by red blood cells, as we've discussed before, carbaminohemoglobin in the red blood cells goes through the body up into pulmonary circulation, released in the capillary bed in the lungs, blown off into, diffuses into the alveoli, then blown off through the bronchioles, bronchus, trachea eventually, and then out. It leaves our body, and that is the process of excretion. There are other things that are excreted. For example, red blood cells, when they are broken down into the body, they produce these bile pigments that are excreted into the GI tract along with bile, and then it goes through the GI tract and it is expelled eventually from the rectum, another way of excretion. However, so much waste products are excreted by the urinary system that often we call the urinary system the excretory system. And those products include ammonia and urea, which are breakdown products of protein or amino acid metabolism. Then there is creatinine, which is a breakdown product of muscle metabolism, and so on. In fact, water is one of the dominant things that are excreted, of course, by the kidneys. And water is a breakdown product of aerobic respiration, just like carbon dioxide is a breakdown product of aerobic respiration. So urine is what is excreted by the kidney. And the kidney itself, of course, there are two kidneys, as mentioned when we talked about the adrenal gland. And like the adrenal gland, the kidney has an outer part, which is the cortex, and an inner part, which is called the medulla. It excretes the urine into the renal pelvis, renal referring to kidney. Then it goes from the renal pelvis into the muscular tube called the ureter. And even here, the ureter engages in peristalsis, which is that concentric sort of muscular contraction to move things along. And so the urine passes down the ureter into the muscular sac called the bladder. And then the muscular sac, the bladder, can push the urine down through the urethra. Now, this is the global view of what occurs, but now we're going to look at the functional unit of the kidney in order to see how urine is actually produced. The functional unit of the kidney is the nephron. The nephron is a filtration, absorption, secretion unit. It is divided into two main parts. There is this part here, which is called the renal corpuscle. And then there's this part here, which is called the renal tubule. The renal corpuscle is composed of a ball of capillaries called the glomerulus. This ball of capillaries has an arteriole that's going towards it and one going away. When it's going towards, it is afferent. When it goes away, it is efferent. So there's an afferent arteriole, ball of capillaries, and an efferent arteriole. Surrounding the glomerulus is Bowman's capsule. Now you have to imagine the relationship between the ball of capillaries, the glomerulus, and Bowman's capsule is as if you took a really soft balloon. You made a fist, and your fist is the ball of capillaries. Then you push your fist into the balloon. Then the balloon will begin to surround your fist. The balloon is like Bowman's capsule, and your fist is the glomerulus. So that is the relationship here. It is at this stage that we have the first step of the function of the nephron, which is filtration. Blood is coming in here, and so it's very dependent on what the blood pressure is, how much filtration will occur. And as blood comes in, then some fluid will end up into Bowman's capsule. But Bowman's capsule will filter out things like cells. The cells are not going to cross. Proteins will be prevented from crossing because proteins are very useful in our blood system, in our bloodstream. And so these things will be filtered, and so other products will end up coming in to Bowman's capsule. Then we have the system of tubules. So tubules are just a series of small tubes. And the tubules can be divided into certain areas. There's this part here, which is a bunch of convoluted tubules. And since they're close to Bowman's capsule, they're called proximal convoluted tubules, as opposed to the convoluted tubules which are further away, which are called distal convoluted tubules. In between the proximal and distal convoluted tubules is the loop of Henle. And there are different limbs or parts to the loop. There's a part that goes down, which is the descending limb, and the part that goes up, which is the ascending limb. And eventually the convoluted tubules end up going towards the collecting duct, which collects the urine. However, as the fluid goes through these tubules, it is always called a filtrate until it ends up in the collecting duct, and that's when it is called urine. Now, in the proximal convoluted tubule, the body will resorb up to 85% of the fluid that just came out into the Bowman's capsule. And in fact, this sort of reminds me of my sister's purse, because when she tries to clean her purse, instead of just removing the things she doesn't want in her purse, she dumps out everything, and then she ends up putting back 85% of what she dumped out. So the body ends up dumping out a large quantity of fluid, and right away it resorbs 85% of that fluid. And this resorption is necessary. It's obligatory, and therefore it's called obligate resorption. So this occurs, and then the filtrate continues along the loop of Henle. In the loop of Henle, this is where the filtrate is concentrated. This is because of movement of sodium and potassium in the medulla of the kidney. There is a concentration in the loop of Henle. In fact, the longer the loop of Henle, the more concentrated the filtrate would become. In fact, I recall looking at electron micrographs, or really close views of the loop of Henle of desert rats. And the loop of Henle is extremely long because for a desert rat, it's best to preserve water, and therefore its loop of Henle is very long to super concentrate the filtrate that it is producing. So here is where it is concentrated. Then it goes on to the distal convoluted tubule. In the distal convoluted tubule, this is where we have facultative resorption, which means we have control over the resorption that is occurring here. There are hormones such as aldosterone that can act here. Aldosterone, as we know, secreted from the adrenal cortex and will resorb sodium, and therefore water will come in passively. So this occurs here. Also, ADH, antidiuretic hormone, can act either on the collecting duct or on the distal convoluted tubule in order to help resorb water into the bloodstream. Then the filtrate passes from the distal convoluted tubule to the collecting duct, which collects the urine and passes it along a system of ducts that ends up going into the renal pelvis, the ureter, and the bladder. The new MCAT changed in 1991 and changed again for 2003 emphasizes DNA and genetics more than any other subject in the biological sciences, and that includes organic chemistry. Now, we've already looked at DNA and mitosis. Now we will look at meiosis and other specific aspects of genetics. Now, in terms of meiosis, it is very important in gametogenesis. Gametogenesis is the genesis of, which means the creation of, gametes, which are the eggs and the sperm. Now, there's something unique about meiosis, and that is that meiosis results in a division and reduction of the number of chromosomes. And there is logic to this, because in mitosis, where we have a certain number of chromosomes, and in humans that is 46, there is a division, and then you end up with the same number of chromosomes. So you have one cell that starts, then you have duplication of DNA through semi-conservative replication, and then you have two cells that result, each of which also have 46 chromosomes. 46 chromosomes in humans is our diploid state. That's the state in which we have 2n, where n is equal to 23. So that's 46 chromosomes, which is the number of chromosomes that is available in almost all of the cells in our body. However, because we have all these different cells dividing with 46 chromosomes, but we are made from one sperm and one egg, so the sperm fertilizes the egg. Clearly, if they're going to add their genetic information together, they must only have half the number of chromosomes that we have in most of our cells. In other words, they must have 23 chromosomes in the sperm, 23 in the egg, so that when it comes together for fertilization, then it creates a situation where there are 46 chromosomes. Then that can divide via mitosis, and then you get tons of cells all with 46 chromosomes. So then you end up with 46 chromosomes. There must be some system in which your cells that have 46 chromosomes could then be reduced to 23 chromosomes to make gametes, eggs and sperm, and then the story repeats itself. So meiosis has two stages. It begins, just like mitosis does, with prophase. So prophase for meiosis is a little different, because in prophase for meiosis, when we start to see the chromosomes condense so that the nucleolus disappears, the nuclear membrane disappears, we start to see the microtubules and the centrioles starting to get ready for the other parts of the cell division. So we start seeing some of the chromosomes, and so they decondense, so that means they become more obvious and more clear. But what's interesting about this first prophase, or prophase 1 in meiosis, is that the chromosomes can cross over, which means parts of the chromosomes literally will cross over another chromosome. Because of this crossing over, these chromosomes can actually exchange parts. So they exchange genetic material, which is very important in terms of understanding diversity and different changes that can happen genetically later on. So this all happens in prophase. Then in metaphase, and this would be, of course, metaphase 1, just like in mitosis, in metaphase, you get lining up on the equatorial plate. Well, here again, in metaphase 1, we get lining up of the chromosomes. But the difference is that the chromosomes that line up in mitosis, each one lines up in mitosis. And then each chromosome is pulled apart into separate chromatids. So that's very different, because then you would have 46 chromosomes lined up in mitosis. But here in metaphase of meiosis, the first part of meiosis, we only have 23 pairs of chromosomes. A pair of chromosome means that one chromosome will line up together with its homologous chromosome, so that you'll get a pair of chromosomes coming together. In fact, there'll be 4 chromatids in each pair. And so sometimes it's called a tetrad, referring to 4. It's also called synapsis. So there is the synapsis of the chromosomes. And then, of course, we have the microtubules that are extending towards, and we have the normal mitotic spindle that exists. But this is all in metaphase 1. Then the next phase is anaphase. And in anaphase, the chromosomes are pulled apart. Now in mitosis, it was chromatids that were being pulled apart. But here, it's actual chromosomes. So you would have the mitotic spindle carrying away a chromosome. And on the other side, you have the same thing, an actual chromosome being pulled away. So this is prophase 1, metaphase 1, anaphase 1. So now you're starting to get reduction of the number of chromosomes. Instead of having 46 in one cell and 46 in another, because actual chromosomes, the entire chromosome, is being pulled away from the other entire chromosome, then you're going to have 23 in one and 23 in the other. This is the reduction division. Then in telophase, this is where you get reforming of the nuclear membrane and the nucleolus and so on. In meiosis, you get a very short interphase. Remember from mitosis, we said P-mati or P-M-A-T-I. So you have after telophase, interphase, and in meiosis, you have a very short interphase, because then you start getting back into the prophase number 2. Now the rest of the cell cycle will be just like mitosis. So in prophase 2, we start to see the nuclear membrane will disintegrate, the nucleolus goes away, the chromosomes condense, and then the chromatids are being pulled apart into separate cells, etc. So it goes exactly like we described for mitosis in the second stage of meiosis. The first stage of meiosis is different, and this is called the reduction division. So it is with meiosis that sperm is created. And the sperm starts off with a primordial cell or a basic cell, which are the spermatogonia, and they divide into primary spermatocytes, which start off the meiosis division, and then the primary spermatocytes become secondary spermatocytes. So they go from 2N to having just N. So going from the diploid state to half of the number of chromosomes, which is the haploid state. And then those secondary spermatocytes will go on to create spermatids and sperm. And of course, sperm have the whip-like organelle called the flagellum. Similarly, gametogenesis in females, which is called oogenesis, begins with the oogonia, which is 2N. The oogonia produces a primary oocyte, which goes into meiosis. During meiosis, the primary oocyte, through a reduction division, because the primary oocyte, like the primary spermatocyte, is 2N, then it undergoes a reduction division and may eventually produce a secondary oocyte. The secondary oocyte, which is 1N, eventually becomes the ovum, which is released by the ovary during ovulation. This is day 14 of the menstrual cycle. The egg then migrates through the fallopian tubes where it may be fertilized. Now, the difference between the production of sperm and egg, one of the important aspects is that when you start with one spermatogonia, you produce 4 sperm. The reason is that you start with one cell that is 2N, it undergoes a reduction division. Telophase, at telophase 1, it's going to separate, and then you'll have two cells. Each of those two cells will undergo a type of mitotic division afterwards. And because it's undergoing a type of mitosis, each one of the cells will produce another two cells. So then you end up with 4 cells from the first spermatogonia. Now, for the egg, starting off with an oogonia, you have the potential of producing 4 ovums, but that does not occur. What happens is that you only produce 1 ovum for 1 oogonia, but you had the potential of having enough genetic information for 4. But what happens is that the 3 other will degenerate. And so the genetic material that could have produced 3 other nuclei will degenerate in just a collection of genetic material which will not be used, and those are called polar bodies. Genetics is the study of the principles and mechanisms of heredity. Central to the discussion of heredity is DNA. Now, we've already looked at DNA. We saw that it is a double helix structure, and we've looked at chromosomes and how each chromosome is composed of 2 chromatids. We saw that this area, the central area, was the centromere, and the distal area was the telomeres. Now, the DNA is wound up using proteins, some of which are called histones. And so the DNA is wound up with proteins to form each chromatid. In other words, each chromatid contains the double helix of DNA. This is why in mitosis the chromatids can separate and yet can still replicate according to semi-conservative replication, because each chromatid will contain the double helix of DNA, which will unzip and serve as a template for new DNA strands to be produced. Now, on the chromosome, as we've already discussed in terms of DNA, there's an area of DNA that codes for 1 protein or 1 polypeptide. We call that area a gene. That segment of DNA is called a gene. The actual location of the gene on the chromosome or on the DNA, the location is called a locus. Now, these are 2 homologous chromosomes. This is sort of a magnification of meiosis, when we saw that the homologous chromosomes will migrate together. So these homologous chromosomes are 2 chromosomes, one coming from your mother, the other coming from your father. So one of maternal origin, the other of paternal origin. And these 2 chromosomes code for the same products. That is the definition of homologous chromosomes. And that makes sense in terms of meiosis, because we saw that the homologous chromosomes will separate in the reduction division in meiosis. So it makes sense because, at least this way, in each cell, you will have enough genetic information to code for whatever products you need. Because right here, you have enough genetic information to code for all the products twice. This has all the information, this has all. So the homologous chromosomes separate. So we have 23 pairs of homologous chromosomes. Now, on the homologous chromosomes, if we were to look very closely, there would be a gene here, for example, and there would be a gene here that would code for the same product. Now, in general, both chromosomes, each chromosome will code for the same product, so there will be a location somewhere where we can say, for product whatever, the size of your ear, this particular product, here is where it's coded for on this chromosome. So they're both coding for the same thing at this particular location, and that is what an allele is. These are two alleles. So they're two genes that code for the identical product. Now, if these two alleles code for the same thing, the same size of whatever, the same color of whatever on your body, if they do the same thing, they're called homozygous. If they code for two different products, then they're called heterozygous. Now, sometimes one of the alleles will be dominant over the other, meaning that if, even though it is heterozygous, one of the alleles is so strong that when you have both of these alleles, one will dominate the others. So that would be called a dominant characteristic, whereas the one which is dominated would be called a recessive characteristic. An example of that would be the color of eyes. One might code for dark eyes, which would be dominant over light eyes. So that if you have the gene for both, your eyes may be dark because the gene for dark eyes is dominant over the other. Of course, this is a simplification. Now let's symbolize the idea of dominant and recessive. Let's say that the gene for dark eyes is symbolized by this, a large E. The idea here is that the capital letters are used to symbolize dominant genes and smaller case letters are used to symbolize recessive genes. So if we look at the different possibilities that there could be, we could have a dominant gene which would be for dark eyes, and if you could possibly have two alleles for that gene, because you would definitely have two alleles. It's either you have one dominant and another dominant, or the other option is you have one dominant and the other is recessive, or the other option is that you have both that are recessive. Now, if you have two dominant, it's clear that you would have dark eyes. If you have one dominant and one recessive, well, then you would still have dark eyes because the dark eyes dominates over the recessive allele. Then if you have both that are recessive, then in this case you have nothing dominating over the other, therefore it would be expressed as though you have light eyes. So then there are two things that are going on here. One is your set of genes. This is described as your genotype. That's your set of genes, either your capital E, your capital E, or your small e. That's your set of genes. The other thing is your phenotype. How are those genes expressed? What do you see on the person? How can you tell? What can you measure? What can you determine? And so that concept is your phenotype. And that is the interaction between your genotype and your environment. That's your phenotype. So when we have these set of genes, we notice that we have 3 different genotypes, but we only have 2 different phenotypes. Either the eyes are dark or they are light. But in this particular example, there's nothing in between. And so we have examples of dominant genes, recessive genes, phenotypes, and genotypes. There are situations in which, because this is one dominant gene, but there are situations in which genes can be codominant. There might be a couple of genes that are dominant over a couple of other recessive genes. An example of incomplete dominance or codominance is seen in the ABO blood types, and we're going to look at that next. As you may know, there are 4 different blood types. They are A, B, AB, and O. So these are the 4 different blood types. In other words, because these are measurable, these are things that we can see about you, these are called phenotypes. So there are 4 phenotypes for the blood types. Now, the way it works is as follows. On a red blood cell, there are antigens, and these antigens are designated this way. So if you have the antigen A on your particular red blood cells in your body, you're blood type A. If you have the antigens B, you're blood type B, AB. If you have both A and B on your red blood cells, then you're blood type AB. And if you have neither the antigens A nor B, then you're blood type O. Now what happens is that these antigens are also called agglutinogens, and that is because if they are attacked by antibodies, the red blood cells will clump, and that is agglutination. So the way it works is that if you have type A blood, therefore you have A antigens on your red blood cells, then in your bloodstream, in your serum, you're going to have anti-B antibodies, meaning you cannot deal with type B blood as a transfusion, for example, because you will attack it and you will have agglutination because you have anti-B antibodies. Of course, you're safe because you have only A antigens on your red blood cells. If you're the type of person who's type B blood, then floating around in your bloodstream, in the serum, is anti-A antibodies. If you have type AB blood, then you have A and B on your red blood cells. Therefore, in your serum, you cannot have antibodies against A nor B, or you would destroy your own cells. And then finally, type O blood. Type O blood, since there is no antigens on the red blood cells, there are antibodies against A and B. Now, you may recall that type O is considered to be the universal donor. Some students like to remember there are two O's in the word donor, so they like to think of it as being related to the type O blood. So type O blood is the universal donor. You may want to pause now and try to consider how is it possible that type O blood can be given to any other person if type O actually has anti-A and anti-B antibodies? Well, the reason is that when blood transfusions are made, the serum is removed. Therefore, type O can be given to any person because the red blood cells have no antigens. So if you give just the red blood cells, not the serum is removed, you give just the red blood cells to a person with type A, no problem. The red blood cells go into the bloodstream and the person does not attack those red blood cells because there's no such thing as an anti-O antibody because there's no antigen on those particular red blood cells. And so for B, you can transfuse type O blood, and for AB, you can transfuse type O blood as well. So, and type O blood obviously can be transfused to a person who has type O blood. So type O is considered to be the universal donor because yes, it has anti-antibodies, but that's removed and the red blood cells have no antigens, so that can be provided to these other people. If you have type O blood, you can only receive blood from someone who's type O because any other blood will contain antigens. And people with type O blood have anti-A and anti-B antibodies, so they will attack anything that provided antigens. So this is how the blood system works, and those are the 4 different phenotypes. Now, in terms of the genes that represent these different phenotypes are quite varied, and you must know that the gene for A is dominant, the gene for B is also dominant. So these are co-dominant genes. The gene for O is recessive. Therefore, in order to have type A blood, you can have, and this is just the symbol for the gene. It's symbolized often with an I, a capital, and also a capital A next to it. So if you have the alleles that have the dominant for A and a dominant for A, so those are your 2 alleles on your particular chromosomes, your homologous chromosomes, then that would code for type A blood. But this also would code for type A blood. If you had A dominant, and then you had the recessive O on your other gene, then that would also code for the same thing. Both of these would code for type A blood. And for type B blood, if you were to have IB and IB, or you had IB and IO as your genes, both of these genotypes code for the same phenotype because the B is dominant over the O. Now, in order to have AB, all you would need to have is the gene for A and the gene for B. And since they are codominant, therefore they would code together for AB. And then for O, well then here, because it's recessive, you'll need to have the gene for O and the gene for O. And so if you have these two alleles for the recessive gene, then they indeed would code for type O blood. Therefore, you have 1, 2, 3, 4, 5, 6 different genotypes to create 4 different phenotypes. So now we can see that in your body, for a particular trait, you may have 2 alleles on your homologous chromosomes. But in a population, there may be multiple alleles for a particular trait, just like we see here. Now, also talking about blood, we do need to mention the Rh factor. The Rh factor is a different gene found on a different locus on the chromosome. So it's not the same as these ABO blood types. So the Rh factor, it's either on or it's off. In other words, you're either Rh positive or you're Rh negative. And the Rh positive is dominant. And in fact, 85% of our population is Rh positive. Now, Rh is different from the ABO blood types, again, for another reason. When you're born with type A blood, you have anti-B antibodies. If you're born with type B, you've got anti-A antibodies automatically. But people who are Rh positive will not have any Rh antibodies. But if you're Rh negative, you might think that you have anti-Rh positive antibodies. Not really. You're not born with them, at least. You need to be exposed to Rh positive blood in order to develop the antibodies against the Rh antigen. Because clearly, if you're Rh negative, you don't have the Rh antigen. So you're not going to have the antibodies yet until you're exposed to the blood. Once exposed to the blood, you develop these anti-Rh antibodies. Then if you get a chance to attack a blood cell that has the Rh positive on it, then it will attack and it will agglutinate. That's why when we say that O is the universal donor, that is true. But it's usually O negative blood that will be provided in emergency situations when someone is bleeding to death or something and there's not enough time to get more specific blood. We will take O negative blood to do the transfusion to provide the least risk of having any reaction against the blood. Now, the time that this becomes a problem is sometimes when a woman is Rh negative during her first pregnancy. If her baby is Rh positive, there might be some exchange of blood during the delivery of the baby. And so the mother might have been exposed to the baby's blood. So there's a little transfusion of blood that occurs there. And because of this, the mother will develop anti-Rh positive antibodies because she's been exposed to it in the baby. Now, if the mother were to have another baby, and if that other baby is also Rh positive, then the mother who has the antibodies against Rh, some of those antibodies may cross the placenta and start attacking the baby. And this would be fatal for the baby if left untreated. Now we're back to our homologous chromosomes. So again, each chromosome in the pair codes for the same products. So if you were to look at the chromosome and you looked at a particular allele on one chromosome, particular allele, and they both coded for the same trait, we now come to our first law in genetics. And that is that if you were to look at these particular alleles that code for the same trait, they are going to end up separating in meiosis, and therefore they will be segregated because each one of these alleles will end up in a different cell because the homologous chromosomes paired and then they were pulled apart. Each will have that particular allele. So this is the Mendel's first law of segregation, and that is that alleles coding for the same trait will be segregated, meaning they'll be pulled apart, put into separate cells. Now, you've got, in humans, 23 pairs of chromosomes. So we have a whole bunch of these pairs of homologous chromosomes, half from maternal origin, half from paternal origin. When these chromosomes separate and go into different cells, it is not the case that 23 from paternal origin will end up in one cell and 23 from maternal origin will end up in the other cell. No, because the chromosomes line up, but they don't say all the guys on the left, all the women on the right. They don't do that. They line up down along the equatorial plate, and then they are separated randomly, independently. Therefore, it could be the case that the one of maternal origin goes left and you look down, maternal origin goes right, look down, left, right, left. It could be in any particular order. And therefore, because they will separate independent of what the other pair of chromosomes does, that brings us our second law. And that's Mendel's second law of independent assortment. Now, given those laws, and of course, like anything else, there are some laws that are broken, and so there are some anti-Mendelian concepts. For example, we expect that the homologous chromosomes will sort themselves independently. But sometimes there's information on one chromosome that sorts independently. Now that is not something that Mendel would have considered. But the reason this occurs is because of what we saw earlier. In meiosis, there's crossing over. So therefore, one would expect this whole chromosome to move over, but sometimes the chromosome will cross over in prophase with its homologous chromosome and they will exchange parts. And so if this chromosome gives up a certain part, then most of the chromosome might go one way while part of the chromosome is on the other chromosome going in the opposite direction. So sometimes information on the same chromosome will sort itself separately. And so knowing that there is this sort of crossing over that occurs, the next question is, well, how can we determine what sort of rate of crossing over is happening? And it has been determined that when you have genes that are very close together, well, then we say that they're highly linked, meaning that the chances that those two genes that are close together on the chromosome or in the DNA, when they're close together, it's highly unlikely that they're going to separate. But if you have genes that are far apart along the chromosome, then the chances that they get separated by crossing over is much higher. Besides crossing over, there is another exception to Mendelian genetics, and that is the idea of sex linkage. Now, according to Mendelian genetics, we expect there to be 23 homologous pairs of chromosomes. So here we have two pairs of chromosomes, and we see that these two pairs of chromosomes have the same sort of look or appearance or morphology. And so we can look at specific areas on one chromosome representing an allele present at a particular locus on that chromosome, And then we can look at the same locus on the other chromosome and look at the other allele. They both might be recessive, one might be recessive, the other dominant, or they both might be dominant alleles. So we expect to see 23 pairs like this. However, there is an exception. There's actually 22 pairs that have the same morphology for all of humans. And those 22 pairs of chromosomes, which would be like this, those 22 pairs of chromosomes are called the autosomes. Then there is another pair of chromosomes, which is called the sex chromosomes. And in females, there are 2 X chromosomes, and in males, there is an X chromosome and there is a Y chromosome. Now, the X chromosome has a similar morphology to other chromosomes, but the Y chromosome is actually a very small, tiny clump of DNA with histones and so on. And so it has an appearance which is very different from the other chromosomes. Now consider this. This has because the morphology is the same, we know that we can find the allele on the other chromosome which is coding for the same genetic information. However, when you have an X chromosome and a Y chromosome, we will see that in males, therefore, there's going to be things that are coded on the X chromosome which are not present on the Y chromosome because the Y chromosome doesn't have as much information as the X chromosome does. So in females, where there would be 2 X chromosomes, then it would be similar to the other chromosomes in which the X chromosome would have another allele on the other X chromosome that will be coding for the same genetic information. So you would have dominant and recessive, and it would be the same situation. However, for males, because there's going to be information coded on the X chromosome, which is not present on that little clump on the Y chromosome, therefore, there will be genes that may be recessive for the female, but for the male, that gene suddenly becomes expressed because there is no other dominant gene in order to dominate or be superior to the recessive gene on the X chromosome. Therefore, that would actually be expressed in the male. In other words, this is the situation where a woman can have a recessive allele. Therefore, because it's recessive, and if the woman is heterozygous, so she also has the dominant gene, therefore the phenotype will not be expressed. The recessive phenotype will not be expressed. However, because of Mendel's laws of genetics, in which those X chromosomes in meiosis will be separated, they will go into different eggs. One egg will have one X chromosome, the other will have the other. Now, in the X chromosome that will have the recessive allele, if that bonds with another X chromosome, in other words, fertilization to create a human, and if it comes together, 2 X chromosomes together, in which one X chromosome has the recessive allele, the other has the dominant, therefore no problem. You have another generation in which that particular woman will be a carrier of the recessive allele, but she will not express the phenotype because she has the dominant allele. Now let's put the situation to the male. Now, if there is the egg with the recessive X chromosome with the recessive allele on it, then it is fertilized by sperm carrying a Y chromosome as the sex chromosome. Then this will create a male. But because the X chromosome carried a recessive allele and the Y chromosome does not contain that allele at all, that recessive allele, which was not expressed in the daughter, will now be expressed in the son. So the son will have the disease. He will have the phenotype. So this is a classic case in which a woman has children, and among her children, none of her daughters have the disease, but all are carriers of the genotype. so they're all heterozygous, then all the sons have the disease. They have the phenotype. So this is called sex linkage. And this is when the disease is linked to the sex chromosomes. Now we've looked at the exceptions for Mendelian genetics. Now we will examine what the consequence is of Mendelian genetics. And the important consequence of this goes as follows. To begin with, recall that capital letters represent a dominant trait, whatever that trait might be. Coding for a particular protein, hair color, nail strength, whatever it might be. So this is a particular trait, and we have it as being homozygous. Homozygous meaning it's both the same, and it's dominant. And this one is homozygous but recessive. Now let's say that the mother had the homozygous dominant trait. The father had the homozygous recessive trait. So it means that the mother, in all her gametes, she's going to have a dominant gene in every one of the gametes, because these will separate independently, and therefore she will always, in all her gametes, after meiosis, she'll have capital A in her gametes. And for the male, he would have the recessive gene in all of his gametes. So if these were to mate, what you would have is, in every single situation, you would have a sperm with the recessive gene and an egg with the dominant gene in every single situation. So all of their children are going to have the dominant A and the recessive A. All of the children will have as their pair of homologous chromosomes for that particular trait. They will have dominant A, recessive A. In other words, they will be heterozygotes, or also we can call them hybrids. So all the children in the population that comes from parents that, as described, they will all be these hybrids. So now, if we were, this particular generation would be called the first filial generation, symbolized by F and 1. So this is the first filial generation. Filial simply means the sons and daughters of the parents. So this is the first generation, sons and daughters of the parents. They would all have this hybrid. Now, if we were to look at the second generation, the next generation, this is how we would do so. We would recognize that in every one, whether male or female, because this particular trait will assort independently, or will segregate independently, because it will do so in the gametes, half of the gametes will have the dominant gene. The other half will have the recessive gene. That goes for male and that goes for female. Half will be each. So if we had a population of hybrids and they were to mate, and you knew that in each of these hybrids they had half their gametes dominant and half were recessive, then what would happen in the next generation of sons and daughters, also called the second filial generation, or F2? So let's examine what would happen in F2. To begin with, we can work this out using a Punnett square. And all that means is we draw a square like so, and now we will put along one side and the other side what the gametes will be, recognizing that we'll put the males down one side, the females down the other. And if we go the males down one side, it's going to be the same thing, which is that each male would have half the gametes would be the dominant A, so one half would be the dominant A, and one half would be the recessive A. And then going along the top, we do the same thing. So 1/2 would have the dominant allele, or A, and the other half would have the recessive allele. Then all we have to do is put in the square what the product would be. For example, the product here would be the product between this and this. So we would have 1/2 times 1/2, which would be a quarter, and we would have capital A, capital A. Down here we have 1/2 small a times 1/2, so that's a quarter, and it would be capital A, small a. And over here, likewise, we have 1/4, and it would be capital A, small a. And then down here we would have 1/4, and we would have small a, small a. So if we were to look at the population to determine what would happen to a population that began with this hybrid or a heterozygote for a particular trait, what would happen to that trait given a population? Well, what would happen is we would get 1/4 of the population with this particular genotype, AA. We would get 1/2 of the population, 1/4 plus 1/4, with capital A, small a, and we would get 1/4 of the population with the recessive genotype, which is small a, small a. So in terms of the genotypes, we would get 1 to 2 to 1, because it's 1/4 to 1/2 to 1/4, so that creates this ratio, 1 to 2 to 1, which would be the ratio of the genotype, which is homozygous dominant, to the genotype, which is heterozygous, and the genotype, which is homozygous recessive. However, in terms of the phenotype, the ratio is going to end up being 3 to 1. And the reason it's 3 to 1 is because what is going to be expressed would be the dominant gene. And so here the dominant gene is expressed. Here it is expressed because even though it's heterozygous and the person would be a carrier for the recessive genotype, it will actually be expressed as the dominant phenotype because it has the dominant allele. So this is going to be the dominant. This would also be the dominant allele will be expressed. So then in 3 cases, you get the dominant allele being expressed, and only in 1 case do you get the recessive allele being expressed. So the ratio for the phenotype would be 3 to 1. The ratios for the genotype would be 1 to 2 to 1, and that is given a population. So in terms of giving a population, you could say, well, there will be a 50% chance that someone's going to end up with the hybrid as a genotype. There'll be a 50% chance given a population. So in other words, you can't say for any particular person being born, you can't say, oh, well, you're going to definitely be this or that. You can say that there's a 50% chance that that person being born will be a heterozygote for that particular trait if both parents were heterozygotes for that particular trait. So it is a probability that you're talking about, not specifically what any individual would have.